This is a coronavirus drifting through your airway. The studs on its surface are spike proteins shaped to latch onto one key target. Some airway cells carry a receptor called ACE2. A single spike reaches out, grabs it, and locks on. The virus is now anchored, but it is still locked out. Then a host cell enzyme called TMPRSS2 can snip the spike like a trigger. The spike springs open and helps drive the viral shell into the cell surface. This is the fast route. The two membranes fuse and a channel opens between the virus and the cell. If surface activation does not happen, the virus can use a backup route. The cell swallows it into a bubble called an endosome. Inside, rising acid and enzymes such as cathepsins can flip the same switch, allowing fusion from within. Either way, the ending is the same. The virus releases a single strand of RNA into the cell. That RNA hijacks ribosomes, the machines that build proteins, turning the infected cell into a factory for new viral parts. Within hours, brand new virions can push out from the cell surface and drift away, each one able to start the process again in another vulnerable cell. 2 ways in, 1 outcome. That is how a coronavirus infects a cell.